In this video, we'll be discussing the European variant of lactase persistence and its possible origins. For years, researchers have debated whether lactase persistence in Europe first emerged from the steppe pastoralists like the Yamnaya or with earlier farming populations of Europe. The Yamnaya theory remains the most popular one till today, because you have probably seen a lot of those content on YouTube where they make edits of Chad pastoralist groups going in different directions and conquering civilizations with a cup of milk in their hand. That's all total nonsense, because a lot of steppe groups do not have lactase persistence, so they are lactose intolerant. They definitely had lactase persistence to some degree, but it was still lower. That, however, doesn't explain us the origin of the European variant of lactase persistence. So we have to go back further. There's another theory that lactase persistence emerged with the early farming populations of Europe, and there are several groups that are considered to be the earliest. Some claim that the oldest known sample having lactase persistence is from Middle Neolithic Scandinavia, which is dated to around 3500 BCE, and others say that it's from late Neolithic farmers in the Basque country, dated to about 3000 BCE. Some even claim that Central European farmers had the European variant of lactase persistence, and they are dated to 5000 BCE. Now this is really old, 5000 BCE, this is actually pre-Yamna sort of. And none of these samples are older than 5000 BCE, but there's one sample that is much older, and I think we haven't discussed it enough. It is an Iranian Neolithic sample from Wezmeh Cave from western Iran. This sample belongs to an individual who lived over 10,000 years ago, and he carried the same European lactase persistence variant that we see in all of those later European populations. And here is what makes it more important, that he had no steppe ancestry and no Anatolian ancestry, and he had no sort of early European farmer ancestry. So we know that this sample predates all of those later groups who are believed to have carried the gene for lactase persistence. I'll explain it to you in a more simple way. We see Iranian Neolithic ancestry in Anatolian farmer group and steppe group, so it makes sense that the Iranian Neolithic population likely had the earliest European lactase persistence variant. So this lactase persistence variant could have spread into Europe through either of these channels. In my opinion, it's likely through the steppe route, because there isn't strong evidence suggesting that Central European farmers had the lactase persistence variant first, but I'm open to both, so it could be both ways. But I think that the steppe route better explains how it may have moved into Europe and got widespread. So what we know is that this Wezmeh Cave sample is the oldest known carrier of the European lactase persistence variant, and that suggests that this trait may have originated further east, in the ancient populations of Iran. Now I will be discussing the Wezmeh Cave sample. Here is a map showing the region the Wezmeh Cave sample is from. The map shows prehistoric Neolithic in Iron Age Zagros genome locations. The next map here shows the modern day peoples with affinity to the Wezmeh Cave sample. As you can see, the groups of South Asia, Iran are more closer to the Wezmeh Cave sample. On PCA, the Wezmeh Cave sample clusters with northwestern South Asian groups like Pakistanis, Afghans, or Northwest Indians. And yes, I forgot to tell you this, that the predicted Y-DNA, or the male lineage of this sample, is G2B2A, and the predicted mitochondrial DNA, or the female lineage, is J1D6. Now, running the Wezmeh sample on G25 calculator, we get these results: 33.2% Setswana, 28.8% Levant PPNB, 14.6% ancient ancestral South Indian. This is interesting to see here. So he has some South Asian hunter-gatherer-like admixture. And lastly, 3.4% African, which is reasonable considering this is an ancient sample and it doesn't have the same genetic drift like West Eurasian groups. If you don't understand what's a genetic drift, you can just comment down below and I can make a video for that, or just explain it to you in the comments. Alright, so moving on. On Eurogene's K13 calculator, we get the results. So this sample is majorly West Asian and South Asian. On Eurogene's K36, he scores South Central Asian for the most part. The closest to this sample, as expected, are Baloch and Makrani groups. Now we discuss the predicted phenotype for this individual. He has a high likelihood of having brown eyes and dark brown eyes, and he had black hair and light brown skin. His hair texture was most likely wavy or straight. He likely had a Greek-type nose and not a snub nose. This is a two-way mix for his predicted phenotype, and based on this we can conclude that he looks very South Asian in appearance. Interestingly, he's predisposed to type 2 diabetes. A similar case is observed in South Asians as well. We see a lot of South Asians having type 2 diabetes, and he has the SCN9A gene, which is commonly associated with South Asian groups. And he's darker in skin color, although first it predicted him to be light brown skin, but he has quite a lot of dark skin markers, so he's quite darker. He's darker than most present-day Iranians. I think he looks like a population from pre-Indus period, majorly Iranian Neolithic-like, with some South Asian hunter-gatherer-like ancestry. This is what the latest paper on Iranian genetics shows him as. He mostly had Iranian Neolithic-like ancestry, with some South Asian hunter-gatherer ancestry. There's another interesting thing that I would like to share. The oldest Indus site found as of now is dated to 7500 BCE, and some suggest it's even older, around 8000 BCE, so Indus is pretty old. We need more excavations done to find what his origin truly is. His origin from Birana is plausible, as he has some South Asian hunter-gatherer-like ancestry, which isn't visible in the Ganjdari sample looking at the current paper on Iranian genetics. So he or his ancestors may have migrated from an early pre-Indus settlement into Iran. This is, however, still a hypothesis, and the more we dig, the better conclusions we reach. The presence of lactase persistence in this sample could possibly strengthen the hypothesis of Proto-Indo-European originating from the northwestern parts of South Asia or Iranian plateau near the Zagros Mountains. I would, however, like to know your thoughts in the comments. Thank you.